Hello everyone, my name is Shubham Mirasgave and today we are discussing about the drug discovery to clinical trials whole process. So first of all, uh, drug discovery. Drug discovery means we have to discover the drug. Uh, so what is needed for drug discovery? First of all, there is a need or demand of any new drug. Then only we have to discover the new drug. Then we have to make concept on ideas, how we can make a drug. There are four phases or types of or stages of drug discovery. Is first of all is target identification. Target identification is to identify the disease uh, for which we have to make a drug target. And then next is a target validation. Target validation means the disease caused on which organ or which part of our body, and that is the target validation. Then lead identification, uh, which source of compound, or we have to identify the lead compound in lead identification, or we can say the raw materials, or from the plant origin or animal origin from which we have to collect our lead identification, means lead compound. After the lead compound identification, phase is lead optimization. In this lead optimization, the we make the lead compound into the medicine form or tablet form or anything, or in the actual drug form in lead optimization. After the lead optimization, we have to trials on the drug. So firstly we try drug on animals, that is called as preclinical trials. In preclinical trials there are studies about the in vivo and in vitro. In vivo means drug is injected in the body, and in vitro means the drug is injected, or drug is applied outside the body. Outside the body means drug is applied on the plasma, tissue, cells and blood, all these things. After the successful preclinical trials, we have to submit INDA form to the FDA for the approval of clinical trials. In the clinical trials, the drug is applied on the human beings, for which we have to get permission. That is the permission is called is IND. There are 5 phases of clinical trials. Phase 0. In the phase 0, there are little numbers or few peoples involved in the microdosing or phase 0, in which in drug is given in the micro form, or 100:1 ratio, and they check the safety of the drug in the healthy volunteers. In the phase 1, uh, there are 20 to 80 volunteers, healthy volunteers, uh, where we check the safety, and the uh, we give the SAD MAD structure. What is the SAD MAD structure? SAD means single ascending dose, and MAD means multiple ascending dose. And after the phase 1, there is a phase 2, which is called as therapeutic exploration, where 100 to 500 patients are involved, for which the drug is applied on them, and or drug is injected on them, and then we just check it out, the how effectively drug is doing his work, or we just in short we check the efficacy of the drug. And after the phase 2, phase 3 is there. And in the phase 3, all type of genetical makeup, all type of human beings are involved. And they drug is or clinical trials are conducted on them. And we check how effective the drug is on all genetic makeup human beings. This is called as therapeutic confirmation. After the successful of phase 3, NDA form, that is new drug application form, is submitted to FDA for the marketing the drug. Uh, uh, there is a 1.5 year. This process take 1.5 year. When NDA form is submitted to FDA, they for the approval of the FDA to introduce drug into the market. Then once we get the permission from FDA, drug is introduced in the market. That is called as post marketing surveillance or phase 4. In this is all about the clinical trials and preclinical trials and drug discovery and development process. Thank you so much.